Hello and welcome to this introduction to the use of different spectroscopic and microscopic techniques to investigate microbial uranium reduction processes. My name is Stefan Hildmann and I'm working at the Helmholtz Center Dresden-Rossendorf at the Institute of Resource Ecology. In this video, I would like to give you a short introduction to the topic of microbial uranium reduction and why this is of interest to investigate. Furthermore, I would like to present you different spectroscopic and microscopic techniques that can be used to get a deeper insight into the occurring processes. Microorganisms can interact with heavy metals in general or radionuclides like uranium in particular via different processes. For example, there can be a bioabsorption of uranium to the cell surface, as well as an accumulation of uranium inside the cells. Furthermore, it's also possible that uranium is complexed by different organic and inorganic ligands. Another interaction process some microorganisms are able to is called biotransformation, which is the reduction or oxidation of the metal ion. Microbial uranium reduction is known for various anaerobic microorganisms, which can gain energy from this process. They are reducing the water-soluble and high mobile uranium-6 to less mobile uranium-4. This process is of special significance for the safe disposal of high-level radioactive waste in a deep geological repository, as well as for new bioremediation approaches. For the safe storage of high-level radioactive waste, it is favorable because in the event of a worst-case scenario, if water enters the repository, the radionuclides can enter the surrounding host rock and then interact with the microorganisms. In this case, a reduction of uranium to the less mobile uranium-4 is favorable because it prevents the uranium from entering the environment or the aquifer. For different bioremediation approaches, it is of interest because the reduced water solubility of the formed uranium-4 simplifies its separation from solution. A currently investigated bioremediation approach is the removal of uranium from contaminated waste waters using multi-species artificial bioaggregates, of which you already heard in the different other chapters of this course. The first step during uranium reduction investigations is the determination of the uranium concentrations in the supernatants, which can be done by different spectrometric techniques as well as colorimetric assays. Decreasing uranium concentrations provide a first hint for an ongoing uranium reduction due to the lower solubility of the formed uranium-4. In general, the proportions of removed uranium from the supernatant are higher than in simple bioassociation experiments, which can be checked by a dead cell reference. Furthermore, fluorescence microscopic images of live dead stained cells can provide information on the cell viability, as well as on a possible aggregation of the cells during uranium incubation. Different studies showed that the uranium-6 reduction is speciation dependent, which means that not every microorganism is able to reduce every uranium compound. Therefore, it is crucial to know the initial uranium-6 speciation in your solution, as well as if it is changing over time. This can be done by different luminescence spectroscopic techniques. At our institute, we are often using the so-called TLFS method, which means time-resolved laser-induced fluorescence spectroscopy. This is an analytical method for luminescent metal ions like the uranium-6. With the help of this method, it is possible to distinguish between different chemical species. The electrons of the metal ion can be excited from ground state to higher energy levels by energy supply. In the case of the laser spectroscopy, this is done by a laser beam. When the electrons are returning to the ground state, they are emitting light, which can then be detected as a sum spectrum of all species present. For uranium-6, you obtain this spectrum with the 5 maxima. With the help of different factor analysis techniques, it is possible to obtain the single spectra of the different involved species. An assignment to different uranium compounds is possible via a comparison with reference spectra. Furthermore, you can also get information about the species distribution of the different uranium compounds over time. UV-Vis spectroscopy can be used to verify the presence of uranium-4 in the samples. This method measures the absorbance of visible and ultraviolet light by a sample. According to the Beer-Lambert law, the absorbance depends on the concentration of the sample, the path length, and the molar attenuation coefficient. Using this method, it is necessary that the uranium is dissolved in solution, which can be done by a dissolution of the cell pellet in, for example, hydrochloric acid. From the obtained spectrum, we can get qualitative and quantitative information on the presence of uranium-6 and or uranium-4 in the sample by comparison of the obtained spectrum with references, as well as on the proportions of uranium-6 and uranium-4 via calculations, including the molar attenuation coefficient. Electron microscopic methods like transmission electron microscopy can visualize the bacterial cells with a high resolution. If this microscopy is coupled with energy dispersive X-ray spectroscopy, it's also possible to obtain information on the elemental distribution in the sample. Signals generated by solid-state electron interactions are used for imaging, and the primary electron beam is generated by an electron source. Using this method, it is necessary to prepare thin-section samples of your uranium incubated cells. This method can answer the following questions. Where is the uranium located and in which amount? Is there an accumulation of uranium inside the cells or just an association outside the cells or on the cell surface? And is there a colocalization of uranium with different other metals, which can provide hints on the possible uranium binding motifs, as well as possibly formed uranium compounds by the cells? To sum up, in this video, we were talking about microbial uranium reduction and its implication on the safe disposal of high-level radioactive waste, as well as on different new bioremediation approaches of contaminated surroundings. We saw that decreasing uranium concentrations in the supernatants provide a first hint for an ongoing uranium-6 reduction due to the lower solubility of the formed uranium-4. We learned about TLFS as a powerful tool to investigate uranium-6 speciation in the supernatants and about UV spectroscopy to verify the presence of uranium-4 in our samples. Finally, we were talking about transmission electron microscopy to localize uranium in the samples if this microscopic method is coupled with EDX analysis. This brings me already to the end of this video. I hope you are now prepared to perform your own microbial uranium reduction experiments. Thanks for watching, and I hope you enjoyed it.